In this video, finally, we can go through the electron transport chain and oxidative phosphorylation. So, oxidative phosphorylation consists of two phases. And we're going to look at some pictures in order to learn this, but phase one is the electron transport chain. That's where we could create a gradient across the mitochondrial membrane. Remember, a gradient is when there's a different concentration of a particle on one side of the membrane versus the other. So in this case, we have a proton gradient, and basically a hydrogen ion is a proton because it lost its electron. So we use this gradient across that mitochondrial membrane, or better yet, I should say, that gradient is created by using high energy electrons removed from our food fuels. And you'll see where these high energy electrons came from, because those were our coenzymes that we reduced in the previous video. Then we go through phase two, which is chemiosmosis. And that uses the energy of the proton gradient, remember, the hydrogen ion gradient, to make ATP. So here's a summary of the process of oxidative phosphorylation, and this is the electron transport chain right here. So in blue here, we're looking inside a mitochondria. This is not a cell, this is a mitochondria inside of a cell. So we're taking just a tiny piece of mitochondria, and that's what we see in this image here. So this is the outer mitochondrial membrane. We have a membrane inside the mitochondrion itself called the inner mitochondrial membrane, or crista is another word for it. And then we have these enzyme complexes embedded within that inner mitochondrial membrane. There's 1, 2, 3, 4 of them, and then we have a fifth one where our ATP is going to be generated. So remember our reduced or high energy coenzymes, NADH plus H, FADH2, that we created in glycolysis and Krebs cycle? Well, now they're in the mitochondria. Some of them were reduced in the mitochondria, and they will go to the enzyme complexes. So complex 1 will take NADH plus H. Complex 2, that's where FADH goes to, this enzyme complex. So these electrons that they picked up during their reduction, they now get oxidized back to their original form. You can see that here and here. We have NAD being formed in oxidation, FAD being formed in an oxidation. So basically it's losing their electrons, and those electrons will travel through these enzyme complexes. So from 1 and 2, they'll go to 3, and there's going to be a coenzyme here that transports or shuffles the electron from one enzyme complex to another enzyme complex. Then you got cytochrome C transporting it from complex 3 to complex 4. So the point here is the electrons are very high energy here from the beginning of this electron transport chain. As they move from complex to complex, they start to lose energy. So we have less and less energy in these electrons as they travel through the electron transport chain. So what this energy does, or what this electron is going to fuel, is hydrogen ions, and I'm trying to find one here. Here it is. Hydrogen ions to be forced out of the space in here, which we call the mitochondrial matrix, out of these enzyme complexes, like a channel. They also act as channel proteins, and into this space, which is called the intermembrane space. Remember, we're still inside the mitochondria. So the electrons being transported acts as a pump to pump the hydrogen ions out of the matrix, mitochondrial matrix, and into the intermembrane space. And at each complex here, 1, 3, and 4, hydrogen ions are being forced out into this part of the mitochondria, the intermembrane space. So what does that do? Well, now we have a high concentration of hydrogen ions, which are positively charged, on this side of the inner mitochondrial membrane, which makes the inside of the mitochondrial matrix more negative. So we produce this gradient of hydrogen ions, which diffusion will say will diffuse in across this membrane into the mitochondrial matrix. So we're pumping them out just so they can diffuse in when we get to this complex 5, which is called ATP, there it is, ATP synthase. So hydrogen ions diffuse back into the mitochondrial matrix, and that will spin this rotor right here, which is an enzyme, which that mechanical action is going to put the phosphate onto the ADP, producing ATP. So this is called oxidative phosphorylation. It's this ATP synthase that is causing this phosphorylation of ADP to ATP. Another important note here is as these electrons get transported or shuffled through the electron transport chain, they're losing energy as they do so, and at the end of complex 4 here, this enzyme complex 4, it's oxygen that accepts the electron. So the final electron acceptor is oxygen. And couple it with some hydrogen, we get water, or H2O, as another byproduct of oxidative phosphorylation. So this figure here gives us the electron transport chain, but instead of structurally showing it how it happens, like in the slide before this one, this shows it to us in energy form by using this graph. So from complex 1, here's complex 2, there's our coenzymes that are giving up their electrons, and you can see as it travels through the complexes, energy is getting lower, free energy in the electrons. And then remember what the final electron acceptor is. That's oxygen, and as oxygens are picking up electrons and combining with hydrogens, there's our water as a, well, I don't want to say a waste product, as a byproduct of oxidative phosphorylation and the electron transport chain. And remember, this whole point here is to pump hydrogen ions out so they can diffuse back in through ATP synthase and create ATP. So this is a good video for you to watch. Of course, you can't click it because this is a video itself, but you have this PowerPoint on iCollege between the welcome module and Unit 1 module. You'll see a PowerPoint module, it's just called PowerPoint, and that's where all the PowerPoints are. So if you go to this PowerPoint slide 44, you can click on this video. It's just gives you a little animation of what's happening to help you understand. So certain poisons work by blocking oxidative phosphorylation. For example, cyanide will block oxidative phosphorylation by binding to complex 4. And remember, that's where the electron will bind to oxygen. So by doing that, we're stopping ATP from being produced through oxidative phosphorylation. And that's how cyanide has its effects. Other poisons called uncouplers abolish the proton gradient. So the inner mitochondrial membrane is permeable to our hydrogen ions. Remember, they get pumped out, and they still get pumped out through the electron transport chain, but they just diffuse back in through the membrane because these certain poisons called uncouplers abolish that gradient. So that membrane becomes permeable. So instead of the hydrogen ions entering through ATPase to produce ATP, they're just diffusing through the membrane, which means the cell cannot produce any energy or ATP. Even at rest, we need about 100 calories an hour or kilocalories an hour. So that places a large demand on our mitochondria, so they continually have to produce energy in our bodies for us to survive. And this is just making the point that even if we're doing nothing at all, we still need this process to occur. And this area down here tells us how efficient our bodies are at producing ATP. So using a mole of glucose the amount of ATP that we produce when taking in consideration all of the kilocalories and what's lost due to heat, well, this what translates to is 38% efficiency. That means 38% of the energy is going toward making ATP, and the rest is heat, which we need in our body. Our most efficient machines can get to about 30% efficiency, with the rest being heat. Usually they're even lower than that, all the way down to 10% efficiency. So this is telling us that our cells are able to work much more efficiently than our most efficient machines that we make industrially. So let's add it all up. Through glycolysis, we net 2 ATP. In the citric acid cycle and aerobic respiration, we net 2 more ATP. Then all of these coenzymes that are entering our electron transport chain through oxidative phosphorylation, we net quite a bit, 28 ATP. We get 10 of these coenzymes times 2.5 per NADH. If you do the math there, that comes out to 25. And then 2 of these coenzymes, FADH2 times 1.5, that gives us 3, so 25 plus 3. End result here, oxidative phosphorylation gives us 28 ATP. So add it up, 28 plus 2 is 30, plus 2 net here is 32. So about 30 ATP is used because we lose a couple of them in a shuttle cost. What that means is, this from the cytosol for this coenzyme to enter the mitochondria takes a little bit of energy to cross the membrane right here. There's the shuttle across the mitochondrial membrane. So when we add all these up minus the 2 right there, we get approximately 30 ATP for every glucose molecule that we began with. Again, here's another good video for you to watch. And once again, you cannot click on this video, but go to the PowerPoint in the PowerPoint folder on this chapter. It's part 1 of the PowerPoint for chapter 24, and this is slide 48. Another good animation to help you understand. So last part of this video, let's look at the difference between glycogenesis, or genesis means forming or creating, so making sugar. Here we have glycogenolysis, so here we're breaking down glycogen. And then we have gluconeogenesis, which means making new sugar. So each one of these is a different process. They look the same as their spelling goes, but be very careful here. There are 3 different processes, so let's make sure you understand all 3. So because our cells don't store ATP, whenever we have high levels of ATP, that's going to inhibit glucose from breaking down. So that's going to promote glycogen, meaning, well, let's store glucose, because if we don't store it, we're just going to make more ATP, and we have a lot in this situation. So we call this glycogenesis, and that just means glycogen is being formed. So glycogen can be formed when there's excess glucose. And that's the storage form of glucose. So glycogenesis is making glycogen, the polymer, out of glucose, the monomer. And this happens mainly in our liver and skeletal muscles, where we store our glucose in the form of glycogen. Glycogenolysis, well, lysis means break down. So this is the opposite of glycogenesis. Glycogenolysis is breaking down glycogen into glucose, into the simple components, the monomers of glucose, the monosaccharide, where glycogen is the polysaccharide. To break it down, we need the enzyme glycogen phosphorylase. And by the way, there is an enzyme, I'll go back, that I missed. In glycogenesis, the enzyme is glycogen synthase when we're making glycogen, but when we're breaking down glycogen, the enzyme is glycogen phosphorylase. So that results in glucose 6-phosphate, which can enter glycolysis in that cell. And remember, in some cells, we can convert glucose 6-phosphate back to glucose, only in intestinal, kidneys, and especially in the liver cells. And when they're able to do that, well, then glucose can then enter the bloodstream and be used by other cells. Now, it's also possible to make glucose from non-carbohydrate molecules. So gluconeogenesis means creating genesis new, glucose, gluconeogenesis. So not from glycogen, because that is a carbohydrate, but we can form glucose from glycerol, which is a lipid, and amino acids, which is in the protein category, from those sources. So when that happens, we call that gluconeogenesis. So when their glucose reserves are depleted, if there's no blood glucose to draw from, our liver will take glycerol and or amino acids and convert them into glucose. So this protects against effects that low blood glucose, hypoglycemia, will have on the body, and which is extremely important for proper functioning of the nervous system. So an excellent summary right here: glycolysis, glucose to pyruvic acid, breaking it down in the cytosol of our cells. Glycogenesis polymerizes the monosaccharide glucose to the polysaccharide glycogen for storage. Glycogenolysis, I'll say that again, glycogenolysis hydrolyzes, breaking down with water, hydrolyzes glycogen, the polysaccharide, into simple components, the glucose monosaccharides. Gluconeogenesis forms glucose from non-carbohydrate precursors like amino acids or glycerol.